Good evening, Samay. Okay, so I am Rahul Saha, and today we'll be talking about the various banding patterns. Okay, in the chromosomes, it has been given in your course the G banding, the C banding, the R banding. Okay, now before I proceed with the banding pattern in the chromosomes, let's discuss the karyotyping. Okay, so karyotyping is a specific technique. Okay, which tells you the you can say configuration of chromosome and in which stage. If we talk about karyotyping, okay, you can say the word karyotyping was derived from the ancient Greek word karyon, okay, which means kernel or seed or nucleus. Karyon. So from here it has come over. Okay. Now chromosomes were first observed in plants. Okay, and the name karyotype was given to it in 1842. There was scientist known as Carl Wilhelm and Carl Wilhelm von Nageli. Okay, this scientist he observed plants and nucleus for the first time. Okay, and then in 1888, Valder named the Carl Wilhelm von Nageli's findings as chromosomes because Nageli didn't know that it was chromosome. He just observed thread-like structure and named it something known as transitory cytoplasts, okay, which were actually chromosomes. Okay, then in 1956, GOJH and Levan found that we humans have 46 chromosomes or 23 pairs with 22 autosomes and one XY/XX, which is the sex chromosome or allosome. So, if you want to define what is a karyotype, a karyotype is a procedure, okay, to study, observe, and examine chromosomes. Okay, in order to find related alteration. Okay, which is referred to as karyotyping. So I'll just show you the karyotype of a Down syndrome, suppose. Then it will be very clear what do I mean by karyotype. Just look at this image. Karyotype of Down syndrome. If you look at a normal karyotype, it looks like this. First pair, the size of the chromosome, the shape of the chromosome will be discerned by karyotyping. Okay, so this is how a normal karyotype looks. Okay, but look at 21st pair. This 21st pair of chromosome, in case of the Down syndrome person karyotype, means an individual carrying one extra chromosome, which I showed you in the last class, that non-disjunction of chromosome in the diplotin stage, leading to non-dissolution of or non-proper dissolution of the synaptonymal complex. Therefore, non-disjunction in the meiotic stage, that would put one chromosome extra in one cell and one chromosome less in the other cell. So if that chromosome non-disjunction happened in the homologous chromosome 21, then the one cell which got one extra chromosome that would have trisomy or 3 chromosome in the 21st pair. So you can see 3 chromosome here. You can see 21 here, only 2 chromosomes. 21 here is 3 chromosome. Okay. Good evening, Pratham. So that is what your trisomy is, and this is revealed by karyotyping. Okay, the technique is known as karyotyping. So the number of chromosomes, the structure of chromosomes, if some chromosome is truncated, half portion is gone, and what kind of chromosomes are these? Metacentric, some metacentric, okay, you can say acrocentric, you can say telocentric. Okay, so all these things, all these kinds of information will be unveiled by karyotyping. So this is what karyotyping is all about, and this is why it is extremely significant as a cytogenetic technique. You are learning genetics via cytology or cell technique or cell analysis technique. So it is known as a cytogenetic technique. Okay. Now what are the steps of a karyotype? If you see or if you have a look at the steps involving karyotyping, first of all you will do what? You will collect the sample. Okay, sample collection. Okay, followed by tissue culture. Whatever tissue you have taken, liver tissue and all, you will be starting to culture them. You will be commencing to culture them via using various media. Okay, RPMI media is taken. Okay, that is not in your course. The types of media taken. Okay, so mostly we take RPMI media, RPMI 1640 mostly. Okay, so it's a growth medium used for cell culturing. Okay, so it is named after Roswell Park Memorial Institute medium. Okay, so that is different thing, that's not in your course. I'm not going to the detail of RPMI medium. Okay, then we have, what we'll do next is we'll arrest the cell or arrest the cells at a specific stage. Which stage? Metaphase stage. Because I have already explained to you that metaphase stage is the highest order of condensation. Okay, highest order of chromosome condensation is achieved in the metaphase stage because you need to divide it. Metaphase plate is produced, right? Then only you can segregate the chromosome later in the anaphase stage. The highest condensation of you can say chromosomes, it is achieved in the metaphase stage, and chromosomes are completely visualized and easily visualized, most easily visualized in the metaphase stage. And our aim is what? To visualize the chromosome. If I arrest it in prophase, it is still decondensing. Sorry, it is still condensing. Anaphase, it will get divided. Okay, so I will not get to see the really the paired chromosomes. Okay, so that is what I wanted to see. In telophase, it will again be, you can say, decondensation or decondensation would again commence. So it is the anaphase stage that would really elude me, and that would be perfect for me to visualize the chromosome. Now the cell, this is a culture medium, so the cell would keep on progressing in the cell cycle. It has crossed G1, S, G2, it has entered M. Under M, it has entered prophase, then metaphase, then it would like to enter anaphase, then telophase also. Now how to arrest it in the anaphase stage? So if you remember in the NEET class, I had talked about a specific inhibitor. There are multiple inhibitors, but mostly what we use is colchicine. Okay, this is the mitotic inhibitor. What it does is it inhibits, okay, the microtubule polymerization. Okay, so this I'll teach you later on in the NEET class. The microtubule dynamics, alpha beta microtubulin, they form the dimerization via, you have GTP attached here, you have GTP attached here. Okay, this one will get dissociated, so this polymerization is stopped. Therefore kinetochore microtubules can never polymerize, okay, from the centriole. which emanates from the centrosome, that is the microtubule organizing center. So this will be taught to you in detail. Just know that without microtubule you cannot have lengthening spindle fiber, and without that you cannot have growth in anaphase, or you cannot proceed in anaphase period. Because anaphase means what? You have the centrioles at the right angle, they will be emanating kinetochore microtubules first, and then polar, and then astral. Okay, the first would be your kinetochore microtubule. So if this is seized, means this will this will reach its length how? This will reach the kinetochore of the centromere how? By polymerizing. So each subunit is alpha beta, then again alpha beta. So if I stop this polymerization, it will stop, or it will cease its growth. So when the growth is ceased, what will happen is you will never ever be able to form a specific microtubule or spindle fiber, that you call it in simple terms, which will be able to reach this specific kinetochore microtubule. If it cannot reach, it will be arrested. Okay, so you have this arrest. Okay, that is known as kinetochore microtubule arrest. Its growth is arrested, seized, and thereby you cannot reach the centromere. Okay, so this is the next step, and then we do what we call separation and spreading of the chromosomes. Third step is, after arresting, separation and spreading of chromosomes, okay, by using hypotonic solutions. Okay, then we prepare, or then we separate chromosomes into a slide. We prepare slides for the chromosomes. We separate chromosomes onto a slide. Okay, the next step is, in order for various chromosomes to be visible properly, okay, this will make it visible. Okay, but after that, if I want to see more of visualization, means I want to see which regions are more high, or you can say highly expressed, which are less expressed, which are heterochromatin, which are euchromatin, then I'll go for staining process. Staining process, and that is only your karyotyping. Okay. That is the karyotyping. So what we have is, you have karyotype generation via this method. Okay. So you can call this the process of arranging, pairing, and organizing chromosomes to find chromosomal variations. That can also be called as karyotyping, right? So we have various ways to do it, and the next stage will be your staining only. Okay, the next stage will be your staining only. One more thing I'll tell you, something known as ideogram. Okay, so ideogram, just like ECG. Okay, ECG is electrocardiograph. That is the graph that we get. Okay, that is the graph that we get. Okay, like this. This is an abnormal ECG. That is the graph that you get. Okay, so same way electrocardiogram is the machine. Okay, so ideogram is the diagrammatic representation of karyotype. Same way ideogram is the diagrammatic representation of karyotype showing all the morphological features of the chromosomes grouped on the basis of position of centromere and ordered in a series of descending size. In a series of descending size. Okay. So that is the ideogram. Means ideogram is this only. Okay, which I just showed you. This is ideogram, the diagrammatic representation of the karyotype. The karyotype is what's being generated, but once you represent it in a diagrammatic way, in order for a scientist or you can say scientist in the labs to interpret, or anyone online to interpret, or any teacher to explain it to you. So this is an ideogram. Okay. So now we come to, you can say, the banding techniques. Okay. So before coming on to banding technique, I think you know all about the various types of chromosomes already. You have metacentric. Okay. We have submetacentric. Okay. Then we have acrocentric. Then we have telocentric. Telocentric does not have any short arm. Okay, it is like this only. Acrocentric will have a very small short arm like this. Submetacentric will have long arm, short arm. Okay. Metacentric will have almost two arms which are equal in length, like this. So this is your simply, you can say, 4 types of chromosomes. Okay. Now one thing I'll tell you, I'll show you, I think. If I type here symmetry of karyotype, asymmetrical karyotype. This is just look at this image. Okay. Just look at this image, that is asymmetrical karyotype. Now when we talk about asymmetrical karyotype, what we mean is, okay, what we really mean is a karyotype showing large differences between smallest and largest chromosome. Between the smallest and largest chromosome of the set and having fewer metacentric chromosomes is called as asymmetric karyotype. Okay, so I'll write the definition first. This is the best definition, asymmetric karyotype. Then I'll describe it. It's very easy. Okay, definition seems a bit, you can say, overwhelming, but it's very easy. A karyotype showing large differences. Between the smallest and largest chromosome. Of the set means of the entire species set means 46 chromosomes or 23 pairs or 23 pairs, okay, and having fewer metacentric chromosomes. Metacentric chromosomes. Now what does it mean? Now come to this image, okay. So if you think of, okay, come to this image, okay, the species of Pinus and Ginkgo biloba, okay, just compare these two. In Pinus, we will see a lot of metacentric chromosome or not. Mostly a lot of metacentric chromosomes you are able to see. Right? This one, this one, this one, this one, this one, this one. Lot of metacentric chromosomes. And also, the difference is also quite less. Difference is, there is not large, there is an absence of large difference between smallest and largest chromosome. You can see. Smallest is this one, largest is this one. So there is not huge difference. There is not a, you can say, considerable difference, a very, very large difference between the smallest chromosome and the largest chromosome. Second point is the Pinus one, it has a lot of metacentric chromosomes, you can see. Both the arms are almost equal. Coming to the Ginkgo biloba, okay, this is a plant, okay. Here you see one sub metacentric only. The rest are, you can say, either, you can say this one is submetacentric, okay, telocentric, submetacentric, and there is a huge difference between the largest chromosome and the smallest chromosome. This is the smallest chromosome. See the size difference, and metacentric chromosomes are very less. Okay, here you can see metacentric chromosomes are very less, and there is a huge difference, a large difference between the smallest chromosome and the largest chromosome. So this one is symmetric, asymmetric karyotype. This one, Ginkgo, and Pinus one is a symmetric one because you have a lot of metacentric and not huge differences present between, or not a huge difference is present between the largest chromosome and the smallest chromosome. Okay. Now, can you tell me how would this relate to evolution? What I postulate is, I say that asymmetric karyotype means Ginkgo biloba. This one is more advanced compared to Pinus. Okay, that is the NEET question. In one of the NEET questions in Cell, okay, 1996, I think, in paper of NEET paper, there was a question. You can find these questions on various sites also. So, what do you mean by more complex or advanced organism? So, answer is asymmetric karyotype means more advanced, not symmetric. Okay, the thing is, asymmetric karyotype means you are more advanced as a species. Means you are more evolved, you can say. Highly evolved, okay, compared to your counterpart. Now a question will directly come into your mind, and must come into your mind, is why so? Okay, why so? So the thing is, you have to go in deep into evolution in order to understand it. So evolution chapter is there in your class 12, little bit is there in class 11th, I think, in biotech part. So the thing is, you can say a nucleus with more metaphase chromosomes appears more symmetric, okay, But the thing is, there is a phenomenon known as scaling. Okay, size dependent, you can say, feedback. So that can be positive or negative. So the negative scaling means when the sizes become shortened. Suppose this was a metacentric chromosome and suppose it has lost some size. Okay, now it has become a telocentric chromosome, or it has become a submetacentric chromosome. So size has shortened. So this is known as negative scaling. Scaling negative means it is losing size. Okay. So this is a kind of trade-off. Trade-off means the lost size will be added to some other chromosome, kind of like a translocation. Okay, this is done. Okay, this is done. This is a deep concept. So I'm just giving you the basics of it. So this is done, you can say, to increase diversity. Okay, because this will do what? If you cut off this gene, because this region will have some genes, you take it off and put it in, suppose this was telocentric, you just increased it little bit, you added this region here, now it has become, you can say, submetacentric. So the genes have got added, so here the genes are getting more diversity. Okay, so you are increasing the diversity by trading off, you can say, right? So this is negative trade-off here. Here it is becoming positive scaling. You are increasing. Okay, again trade-off. So you are trading parts of chromosomes. So that keeps on happening when you have to increase diversity. Why? Because you need to adapt. So evolution means the ability to adapt as a species, and more the ability to adapt, better you have the chances of survival. Your likelihood or probability of survival increases that many folds. That is evolution's real meaning. Evolution doesn't mean that you are the best. Evolution means as per the current habitat, you have been able to adapt. So it is nothing but adaptation. But adaptation will happen how? If the genes acquire variation. Variation means change in nucleotide sequences. So that can be done like that. If you mix two genes, if you vary genes, you cause frame shift mutations, SNPs. Okay, so gene variation will lead to difference in nucleotide sequences, that will lead to difference in the protein formed. So the protein's conformation will change, or simply I can say protein structure will change. So when structure will change, function will differ a bit. Okay, function may improve, or in some cases may regress also. It's not that every time variation is good. It may be an unfavorable variation also, but mostly it is a favorable variation. So function will improve, and therefore your trait or character will improve, that you call in genetics. Okay, and you will be able to adapt. Okay, suppose you need to sweat lesser in order to conserve body fluid. If you are living in a very hot area, okay, if you sweat profusely, you will dehydrate. So there the proteins, your NAK ATPase shall generate lesser amount of energy. Okay, because more that will cause more body heat production, because NAK ATPase, what it does, thyroxine hormone will stimulate this pathway, where I have told you that Na plus is always more outside the cell, Na plus is less inside the cell, K plus is more compared to K plus concentration. Okay, I'm not comparing Na plus K plus. K plus concentration is more compared to K plus concentration, which is less outside. But Na plus K plus ATPase, it throws 3 Na out and 2 Na in. You can see it is doing it against the concentration gradient of the ion. Okay, it's not letting Na come in. It is throwing Na plus out even though Na plus is lesser inside, and throwing K plus in even though K plus is more outside. So this will require a hydrolysis of ATP to release energy. So energy will be used to go uphill against the gradient, and the remaining energy would increase the body heat. Okay, so these are the conceptual things about karyotype that you should be knowing in order to solve any kind of NEET questions. Symmetry, asymmetry, which is more advanced, which is more evolved, that is directly asked in NEET syllabus. Okay, So now I have also prepared huge, you can say, your worksheets which are of 140 150 questions. Okay, so now I have prepared that also. I'll be sharing that with you in the next NEET class. Directly 140 questions or more than 100 questions at least pertaining to one chapter, comparing of all the previous year NEET questions. Okay, and once we are done with that, then we'll switch to, you can say, probable questions that can come, okay, irrespective of the previous year. But first let's finish the previous year question, that you may get acquainted with the syllabus and how the NEET questions are asked. Okay, so next we come to types of staining. Okay, so we have various types of staining, or simply known as chromosomal banding. Okay, it is given in your book, chromosomal banding technique. Okay. It was first shown in 1971 conference in Paris and is defined as, okay, if you want to define it, a part of a chromosome which is clearly distinguishable from its adjacent segments. By appearing darker or lighter with various binding methods. Okay. So this is the definition of chromosomal or chromosomal banding techniques. This means what? If I show you an image here, chromosomal banding, or directly if I show you G-type banding, which is one of the chromosomal banding images. Okay. So light and dark. If you see here, okay, just take it, this one as dark, this one as light. Okay, this is kind of orange, you can take dark, you can take blue as light band. Okay, that just take it like that, that orange is a dark band, means it looks dark when you visualize via, or when you visualize it under the microscope or under a microscope, and the lightly colored, you can say, sky bluish one is the lighter one. Okay, so if you want to divide chromosome into dark and light bands, okay, that is known as banding pattern or banding technique. And why do we need to divide? Because we need to see where are AT rich regions, where are GC rich regions, in which part of the chromosome is heterochromatin mostly present, facultative, then constitutive, in which part of the chromosome euchromatin is present. Okay, in order to see all that, which part of the chromosome is gene rich, right? Because 98.2% is gene poor. So which part of the chromosome is gene rich? So 1.2 to 1.8% of the chromosome is gene rich, more than 98% is gene poor, that I have already discussed with you. So exactly which part of which chromosome, because we have 46 chromosomes, so each chromosome will have different number of genes. So which part of which chromosome has more gene, which part of which chromosome has less gene, which chromosome is densest when it comes to gene number, which chromosome is rarest or possesses the least number of genes, which chromosome possesses the maximum number of or amount of or maximum region of heterochromatin, that is the non-genic region. So all these critical data will be analyzed and will be revealed after banding only, because dark and light regions will be generated. Mostly what happens is dark regions are gene poor regions, light regions are gene rich regions. You can see dark region is more in number and they occupy more. So dark region is gene poor, light region is gene rich. So now we'll come to the bandings that are given. Okay, little bit of history is there. In 1958, okay, scientists like Casperson, they published that the use of, you can say, quinaracrine mustard stain to study or to unravel or to usher a new area of chromosomal banding. And in 1971, which was I was talking about, it was the first attempt to provide nomenclature for chromosome banding in any species. Okay, so it does allow us to visualize smaller pieces of the chromosome so that we can identify smaller structural chromosomal abnormalities also, which are not visible on a, you can say, or via any routine abnormalities or routine, analysis. Okay, now we have various banding techniques. Okay, we have G banding, we have R banding. Okay, talk about banding. Okay, we have G banding, we have R banding, we have Q banding. Okay, then we have NOR banding also, nucleolus organizing region. Then we have C banding also, which is the centromeric one. Okay, so G and R, they are very, you can say, similar. R is known as reverse G banding, so sometimes it is not included. Okay, so G banding was discovered in 1971. Okay, Q banding, which uses the dye quinaracine, or I can call it quinarcin. Okay. G uses the dye known as Giemsa stain. Okay. So this was discovered in 1971. This was discovered in 1958. The oldest one is this one, Q1. Then 1973 came this one. Then 1978 came this one. Okay. Now what happens is, when we go for Q banding pattern, okay, each banding I'll tell you. Q banding. So we take chromosome at the metaphase stage. These are given in your book, different banding techniques. Then we stain it with quinarcin mustard stain. Okay. Then it is subjected to UV light. Okay. Then we generate the banding pattern. So mostly what will happen is the quinarcin will denature the DNA and UV light will denature it more. Okay, and then banding pattern will give rise, means the staining will be done more in the AT rich area. Okay, AT rich region. And this is GC rich region, you can see. An AT rich region and a GC rich region. So what happens is, so chromosomal banding, okay, the number and size of bands, okay. So the bands are number, or the total number of bands, they will be resolved, okay, after the banding pattern is done or after the banding is done. So AT rich region will be known as your gene poor region, or simply it will be known as heterochromatin. It will quench the dye more and it will fluoresce more. Okay, it will show more fluorescence, and it means heterochromatin. More fluorescence means it will quench the dye will bind to AT rich region more. Okay, and this will lead to more fluorescence in the AT rich region. Okay, now next thing is GC region will not fluoresce that much. It will not fluoresce that much, less fluorescence or may not fluoresce, and because of less fluorescence or absence of fluorescence, that will be known as euchromatin or gene rich region. Euchromatin or gene rich region, because this dye has more affinity to bind to the AT rich region than to the GC rich region, and AT rich region is really gene poor, and you can say GC region is gene rich. We have also CPG islands, okay, where G is followed by C, okay, that is more of gene rich region. Okay, in the promoter region it is found, right? So this is your Q strain. Okay, then coming to the G staining, or Giemsa staining, and I'll also explain to you R staining or reverse Giemsa along with G staining technique. Okay, so what happens in G staining is we first stain with, in G staining what we do, not R staining, G staining, we add trypsin. We treat the DNA with trypsin. Okay, it is treated with trypsin. Okay, then it is a protease treated chromosome. Once it is a protease treated chromosome, what happens is then it is treated with Giemsa. Okay, the Giemsa stain. Okay, sometimes urea can be added, but then it is treated with Giemsa staining or Giemsa stain. Again, what happens is the AT rich region, okay, and you have GC rich region. So AT rich region will be stained dark, means more staining will be done. Okay, in GC rich region less staining will be done, so it will stain light. Okay, again gene rich and gene poor region. But in case of R staining, it will be opposite. It will be opposite because the R stain will do what? The R stain will completely denature the AT region. How? Because AT is double bond, GC is triple bond. Okay, so it's easier to denature AT, but R staining, that's why it will denature AT completely, because it will use, you can say, hot acidic saline solution and then G staining. So in R staining, what we will do is, in R staining technique, we will first use hot acidic saline solution. This will denature AT completely. Okay, AT denatured completely. A double T bond will become A separate, T separate, but not able to denature GC completely because of its triple bonding. Okay, so now what will happen is, it is a reverse band and then Giemsa staining will be done. Then I'll add Giemsa stain. Okay, so now what will happen is, where denature has not happened properly, GC, it will stain it dark. Where denaturing has been happened completely, then or you can call it under denatured, and here it will be able to bind in a very less manner. So this will be dark band, this will be light band. Just the opposite. Reverse Giemsa staining is R staining. Now coming back to Giemsa staining, what happened in Giemsa? So G banding is, first I added trypsin, okay, then I added Giemsa stain, and then what I got is GC80. Now I am saying that this happened here, dark stain happened, here light stain happened. Why so? So it depends upon the denaturing capacity, okay, depends upon the denaturing capacity. Yeah, I know it is little bit boring, but it is there in your book. Very, it is, you can say, conceptual, okay, it's a conceptual topic, so, and it is there in your course also, so you have to study it, and it is very technical. Just go through the technicality of this. Just see, trypsin I am saying. So how will trypsin denature the DNA? Okay, how will trypsin denature DNA? Impossible. What it does is it lengthens the histones. It denatures the, you can say, various peptide bonds in the histones. Okay, it will break or it will reduce the disulfide linkage in histone, and then it will break the peptide bond in the histone surrounding the DNA. So in which part is more histone present, AT part then GC part? So GC will be more denatured because less is there, because here less histone is there. AT will be, GC will be, you can say, more denatured, and where denaturing is more, there it doesn't bind properly. That is the trick. So learn via conceptuality. Okay, where denaturing is more, there the stain would not bind because it is already denatured. Okay, it binds to double stranded DNA. Okay, it binds to DH DNA. Here what will happen? More histone is there, so less denaturing will happen of the DNA. Therefore more binding will happen. Okay, so therefore it will stain dark and it will stain light. Okay, that is how I'll get, you can say, the, you can say, heterochromatin and euchromatin region, or gene rich and gene poor region. Okay, so then we'll come to one good question comes, why is G banding not used in plants? Because human mitotic chromosome, G banding, again a good question of NEET. Why it is not used in plants? Because human mitotic metaphase chromosome, is only 2.3 times shorter than the main chromosome, that is the condensation, whereas plant mitotic chromosome is 10 times shorter than its prophase chromosome, you can say. Ten times shorter compared to the human counterpart, human chromosome. So since it is so short, lesser genes, it becomes extremely difficult, okay, to go for G banding technique and get proper result. Okay, then you have something known as NOR banding technique, or N banding it is known as. I'll just go through it quickly. Okay. NOR banding, nucleolar organizing region. I told you there are 10 chromosomes, so you can have 10 nucleoli. Okay, last class. So we take chromosome Two more banding are remaining only. Then from next class we'll switch to a very interesting topic. Okay. Then this is air dried. Okay. Then it is treated with 5%, you can say, trichloroacetic acid. Acetic acid at 95 degrees Celsius for 30 minutes. Okay. Then it is treated with .1 normal HCl at 60 degrees Celsius for again 30 minutes. Then what you get is the banding pattern in you can say in the structural non-histone proteins linked to the NOR region. You get a specific banding pattern of that region. Okay. So that is known as our NOR banding. Okay. So NOR banding technique is a very, very special technique. Okay. It uses silver stain also sometimes that preferentially accumulates in the nucleolar organizing regions. Okay, which are the satellite DNA regions of the acrocentric chromosomes. Okay, so that is also used, okay, in the nuclear organizing region. Okay, to know where the nuclear organizing region is present. So it also uses ammonium silver nitrate solution. Okay, that can also be used, ammonium silver nitrate solution here. So this can also be used, silver nitrate solution. Okay. Then the last method I'll come to is directly the C banding technique or centromere banding technique. That is the last topic. Okay. Here chromosome is treated with an alkali solution. Okay. Then it is, this will, alkali solution will denature the DNA again. Okay. Centromere is highly associated with histone, so it will get less denatured. Okay. Then it will be washed with sodium citrate. Okay. Sodium citrate at 60 degrees Celsius for 30 minutes. Okay. This will cause the renaturation of only repetitive DNA. Very, very important, because centromere is a repetitive DNA. So renaturation of repetitive DNA will happen, but renaturation or rebinding of non-repetitive DNA will not happen. Okay. Then it will be stained with GIMSA. Okay. Then you can get heterochromatin region easily. Whatever has renaturated will be stained and will be dark. So heterochromatin region will be seen. So this is your centromere banding pattern, or simply C banding pattern. Okay. So this will cover all your banding techniques. Okay. And each chromosome has, I'll tell you, one long and one short arm. Okay. Short arm is known as P arm, petite. Okay. Long arm is known as Q arm. There is no full form of Q because Q comes after P, so it is known as long arm. Suppose this is a submetacentric chromosome. Okay. One more thing I'll tell you is, okay, Sometimes this Q is known as Q. Okay, QUE. Okay, sometimes it is called as Q as full form. Okay, these are the cytogenic bands, and sometimes the bands are labeled as P1, P2, Q1, Q2. So first band, second band here like this, first band, second band here like this, it will be labeled. Okay. So suppose nomenclature of chromosome, that is very important. So if you are given a cytogenic map and something is written like this, suppose a location of a gene is located at this location 7q31.2. What does this mean? Suppose this is the address of the gene. So what does this mean? Chromosome number 7 is located on chromosome number 7. Okay, which arm? Q arm. Q arm is the Q arm or the long arm, right? Then band 3, which band? Third band. Suppose this is the third band, band 3. Okay, then sub band 1. Under this also you can have sub band. So sub band 1. Then under that also you can have sub band. So 3.1.2, that is the 2 is sub band. Sub band. Okay. 2. So this is how you locate a gene. That is the meaning of 7q. You will see in a lot of research papers in your some books also. You will see 7q31.2. That is the meaning. 7 chromosome, Q is the arm, then 3 will be the band, then 1 will be the sub band, and after point you will be having sub band. Okay. So I hope you were able to comprehend the lecture. If you have had any doubts, you can ask me on my personal number. I'll help you out as much as possible. So next class we will be starting a new topic, a very interesting topic. Okay then. Thanks, Samesh.